Hello everyone, and I am Anupama, and welcome to the problem solving class of Human Molecular Genetics course. Today we will be solving some problems related to pedigree, but before we dwell further in the topic, I hope you must have seen first and second lecture of week 2. I hope explanation while solving the problems will help you build your concepts and make it more clear. Now, not wasting much of your time, let's do the first problem. So, our first problem for today is to calculate the penetrance of the disease in the pedigree. Now, to understand this problem, we need to know what is penetrance. So, it so happens that in many diseases, even though the individuals have the genotype related to the disease, phenotypically he or she is normal. That is, you have the disease causing gene, but you don't have the disease. This is called incomplete penetrance. Having learned that in this pedigree, we need to find out individuals with the disease that are phenotypically showing it and the ones who have the genotype for it. Now let's see which all individuals have the disease. Now that's an easy thing. We can just count the number of individuals whose boxes are filled. In this case, the red ones. So there are 5 individuals which have the phenotype for the disease. Now we have to look into the genotype. So if you see this pedigree in the 3rd generation, 1 and 3 they have the disease, however neither of the parents have the disease. So it seems that individual 2, 1, that is this individual, should be carrying the genotype for the disease, however he is not manifesting the disease phenotypically. So the genotype for this person should be A and A, as his parents 1 and 2, out of the 2 has the disease. So this brings us that there are 6 individuals who has the genotype, but out of 6 only 5 are showing the disease. Now if you divide 5 by 6 and multiply it by 100, then this is the penetrance for the disease. So this is how we calculate penetrance of any particular disease. Now let's look into the second problem. The second problem is a bit big, and I'll go through it. Maya has just learned that she has adult polycystic kidney disease. Her mother also has the disease, as did her maternal grandfather and his younger brother, both of whom are now dead. As far as Maya knows, no one else in her extended family has the disease. Although she had a sister, Ahilya, who died in car accident when she was 16 and might have showed symptoms if she had lived long enough. Maya is 42 years old and has 3 children with her husband Aryan. Ananya is 20, Anamika is 18 and Tarun is 15. So this is the question which is being shown in front of you. Now there are few problems related to this pedigree. So we'll go one by one to all the problems. Now our first problem is draw a pedigree for this family using the proper symbols. So let's draw it. Here we got the information about this family through Maya. So Maya is a proband and she is female. So let's draw a circle for her. And as she was diagnosed with polycystic kidney disease, so we will fill the circle. Now let's draw Maya parents. As we know from the problem that her mother also has the disease, so let's draw her parents. This circle denotes her mother and she had the disease, and this square denotes her father, as did her maternal grandfather and his younger brother, both of whom are now dead. So Maya's mother and now her parents, that is Maya's grandparents. So this symbol, that is square, denotes his grandfather, grandmother, and now her grandfather's younger brother also had the disease. So, we will fill the circle and both are dead. Now, let us come to Maya again. As far as Maya knows, no one else in her extended family has the disease, although she had a sister Ahilya who died in a car accident when she was 16. So, Maya's sister Ahilya, she died when she in a car accident. So, we will just cross. And now, let us look into Maya again. Maya is 42 and she is married. So, this is Maya's husband Aryan, and she has 3 children, that is Ananya, Anamika and Tarun. They are young, so we do not know that whether they would have the disease or not. So, this is the pedigree which we have drawn for Maya. Maya is the proband, so we will make an arrow, and let us now denote the generation. This is first generation, second generation, third generation and fourth generation. So, this is how we draw pedigree. Second question, and I have shown the pedigree for the reference to answer the questions which would be coming ahead. So, the second question is, could this disease be autosomal recessive? If so, is this mode of inheritance likely? Explain. So, like looking into the pedigree, if we see almost all the generations have the disease. So, it does not seem that this is a autosomal recessive disorder. It seems that it is a autosomal dominant. But let us look into what are the possibilities through which we can explain that this mode of inheritance can also be a autosomal recessive one. So, if it is a autosomal recessive disorder, then if you see 2, 2, that is this one, this person. So, to have a disease in second 2, and if it is a autosomal recessive disorder, then 1 should also be a carrier. And similarly, if 3, 2 has the disease, then 1, second 1 should also be the carrier for the disease. But it is very unlikely that both of the persons, that is 1, 2 and 2, 2 are marrying to a carrier. So, it does not seem that this is an autosomal recessive, and hence it is a unlikely possibility which I have written, that this disease could only be autosomal recessive if 1, 1 and 2, 2 are carriers of the disease, which is rare possibility. Thus, it is unlikely that this disease is autosomal recessive. Now, let us look into the third question. Third question is, could this disease be autosomal dominant? If so, what is the chance that Anamika will get the disease when she reaches middle age? Now, suppose second daughter of Maya is Anamika, that is 4, 2. Coming to the first question, it seems likely, very likely that this is a autosomal dominant, because if you must have gone through lecture first of week 1, where we have explained what is how does the pattern of autosomal dominant is, that in all the generation it shows, and it affects male and female equally. So, it seems that very likely that this is an autosomal dominant. Now, what is the chance that Anamika will get the disease when she reaches middle age? Now, Anamika's mother Maya, at age of 42, she has developed the disease. So, she must be having, her genotype must be, if we denote capital A and small a for her genotype, then it is likely that Anamika would have 50% chance, as one of the gametes will be coming from Maya. So, it seems that 50% chance is for Anamika to develop the disease. Now, that is what I have written here, which you can very well read, that this disease shows the classic pattern of autosomal dominant inheritance as it is seen in all, and Anamika has 0.5%, that is 50% chance to develop the disease. Now, let us look into question number 4. Could this disease be X-linked recessive? Explain using specific individuals in the pedigree to support your answer. So, what are the rules, like if it is a X-linked disorder, how the pedigree should be. Now, if you see, it is coming from 1 2, that is male, to female, well taken, and maybe one is carrier. But had it been autosomal recessive, then 2 1 should also be showing the disease, because this 2 1 is male, and for X linked recessive it shows in the male. So, it does not seem likely that this could be a X linked recessive. So, that is what I have written, that no, had it been X linked recessive disease, father of 3 2, that is Maya, that is 2 1 should have shown the symptoms of the disease, which is not the case. Now, let us look into the fifth question. Could this disease be X linked dominant? Explain using specific individuals in the pedigree to support your answer. Now, again, if it is a X linked, then it passes from male to female, and from female to both the genders, like to her sons and daughters. So, that is what is being seen. So, it can be, but this mode of inheritance is uncommon, but there is nothing in the pedigree that rules it out. So, that was with this pedigree, that was pedigree problem 2. We solved 5 questions and we ruled out why this pedigree is not X-linked recessive or autosomal recessive and X-linked dominant, and most likely this is a autosomal dominant disease. Now, this was a problem which was related to a particular pedigree. Now, there are problems to a population too. Now, let us look into such problems. That is our pedigree problem 3 for today. So, let me read the question for you. An autosomal recessive disease, supposedly Lafora disease, affects one newborn in 10,000. So, what is the expected frequency of a carrier in a population? Till now, the problems which we were dealing was with the particular pedigree, but now we have come to a population. So, before we solve these problems, there are few assumptions which are required to be made, like in mathematics we do assumptions. So, in genetics also, to solve a problem related to a population, we need to make some assumptions. So, what are the assumptions? The assumptions which need to be made are that population size is infinitely large, that is, it is quite a large population. Mating is random, that is, it is not specific that only a particular community will be the one marrying each other. That would change the frequency of the alleles. Now, allele frequency are equal in the sexes, that is, there is no sex biasness, that this particular allele will only be in male or this particular allele will only be in female. So, not such biasness should be there. There is no migration, mutation or selection, that is, in the population is neither migrating, nor there are mutation, nor there are selections. So, why are we making these assumptions? Because we need to calculate a frequency for a allele. So, we are defining the boundaries for the populations. So, these assumptions if you take these assumptions, this was proposed by Hardy-Weinberg, and this is the equilibrium. So, it was proposed that if there is a allele E, A, and its frequency is P, similarly there is another allele A and its frequency is Q. Now, if you calculate the genotype frequency in the same population, the population with these assumptions, then a homozygous AA will have P square, a homozygous small aa, that is Q square, and the carriers, that is capital A and small a, would be 2pq. Having understood this, so there are two equations which we have, like for allele frequency p plus q is equal to 1, and for genotype frequency it is p square plus 2pq plus q square is equal to 1. So, these two equations will help you solve problems related to population genetics, considering the above 4 assumptions. Now, let us look into the problem which was before us. So, this is an autosomal recessive disease, Lafora. Now, let us look into what would be the genotype for unaffected and affected. So, genotype for the unaffected would be capital A and small a, and capital A and capital A. Since this is a recessive disease, so it would be only manifested into individuals who carry small a and small a. So, and the frequencies, if we write, this would be p square, this would be q square, and this would be 2p q. Now, we know from the problem that recessive disease, the q square is 1 by 10,000. And we have to figure it out what is the frequency for the carriers, that is capital A and small a, that is we have to calculate 2pq. To calculate 2pq, we need to know what is t here. So, if q square is 1 by 10,000, so q would be very simple, 1 into 100. Now, as you remember, p plus q is equal to 1. So, if q is 1 by 100, then p would be 99 by 100 to make it 1. Now, we need to calculate 2pq. So, 2 into 99 by 100 into 1 by 100. Now, if you multiply, this would come around 198 upon 10,000. Approximately, we can say 200 by 10,000, which would be 1 by 50. So, we know that the expected frequency of the carriers would be 1 by 50, that is every one individual in 50 would be carrier for this disease. Now, let us look another problem which is related to the same. Now, if a parent of a child affected by the earlier disease, that is Lafora, remarries, what is the risk of producing an affected child in the new marriage? Let us make a simple pedigree here. So, we have a affected child, we do not know the gender. So, I will just make like this, and this is affected. And these two are his or her parents. Now, one of the parents decides to remarry. Suppose mother remarries, and now they are willing to have another child, gender we do not know, but what is the frequency or what is the chance for producing an affected child? Now, since she had a child which was affected, so she must be a carrier for this disease. Let us write A. Now, we need to know what is the frequency of, or what is the chance of this person to be carrier for the disease. Now, as we calculated earlier, the chance for this person to be carrier, that was capital A and small a, if you remember, it was 1 by 50. And if both are carriers, so let us draw one small Punnett square. Now, you see that this is 1 by 4th chance for the child to be small a. So, to calculate what is the risk would be 1 by 4 into 1 by 50, because this person to be the carrier is like if this person is carrier, this is 1 by 50th chance, and if he is carrier and she is carrier, then this is 1 by 4th chance for the person, for the child to be affected by the disease. So, this brings us to 1 by 200. This would be the risk of producing an affected child in the new marriage. Now, let us look into problem 5. Now, what we learned with Hardy-Weinberg equilibrium, there we assumed that every gene has two allele, A and A. But there are some cases, like if we consider male, for X they are hetero, and for on this on the other hand, females, they have 2 X chromosomes. So when it comes to male, the equations will change. There will only be one allele, either capital A or small a. However, in the females, the genes which are present on X, they will follow the similar one, that is q square plus 2pq plus q square is equal to 1. Now let us look into the problem. This is a X-linked red-green color blindness, affects 1 in 12 males. What proportion of females will be carriers and what proportion will be affected? So what we need to calculate here, that what proportion of females would be carriers, that is capital A small a, and what proportion would be affected, that is small a small a. Now as I told, for male, since there is only one X chromosome, so either there will be capital A or small a, and from the problem we know that 1 in 12 males, that is the frequency of A, and if I say that is Q, it is 1 by 12. Now if I have to calculate P, going back again, P plus Q is equal to 1. So if Q is 1 by 12, then P would be 11 by 12 to make it 1. So the frequency of P would be 11 by 12. Now I need to calculate carriers, and if you remember, the carriers would be 2pq. So this would be 2 into 11 by 12 into 1 by 12, which would 2 into 11 would be 22, and 12 into 12 would be 144. So the carriers, the female population would be 22 by 144, that is out of 144 females, there would be 22 females which would be carrying this disease. Now we need to know what is the affected portion of the population, that is AA, and that would be Q square. So Q square would be 1 by 12 into 1 by 12. So this gives you the frequency for 1 in 144 females would be affected by this disease. So that's all, and I hope going through this class would help you solve some problems, and I might come up with some more problems and help you in solving them. Thank you.